Hello, everyone. My name is Angie Jaramillo. I am a student at Mercy College, and my research is understanding how Oomycete plant pathogens interfere with the immunity of host plant Arabidopsis thaliana using bioinformatic and virtual tools. So plants, they lack an adaptive immune system and instead rely on an innate immune system that is just consistent of systemic signals that come from the infection site. So, effector proteins are molecules that are secreted by pathogens that cause disease in host plants and can lead to death. And these effectors activate hormonal pathways. For example, my research highlights the JA pathway, which we can see here in figure one. And the pathogen I am looking at is Oomycete, which is a fungal-like pathogen. Specifically, the Oomycete I'm looking at is P. sojae, which is a causant of the stem and root rot of the soybean. So my hypothesis is that P. sojae will actually suppress the defense mechanisms of the JA pathway. So what I did was I performed a literature survey to identify the JA pathway in plants. From here, I then created a model of this JA pathway using Plant SimLab, a virtual lab simulator. I performed some knockdown experiments we can see here in figure 3A, and I identified three defense genes that are critical, which is COL1, JAS1, and JAS3, and PDF 1.2. And then I performed a suppression assay. I infiltrated plants with my effector protein, which is AVH73, and a buffer. And I also performed some RT-qPCR by extracting RNA. So we can see here some results of the infiltration. 4A is the buffer Brugmansia that I infiltrated, and 4B is the effector Brugmansia that I infiltrated. We can see the score sheet over here. And just to highlight that we can see that the effector Brugmansia is definitely much more like hit of infection. We could see browning of the stem. You can see how dry these leaves look, browning of this. Crisp, it's just a crispy, dry feeling, and versus what a healthy Brugmansia leaf would look like. And here are my RT-qPCR results. I tested JAS1 and PDF 1.2, and we can see here that AVH73 does suppress JAS1. The p-value was 0.01559, which shows statistical significance versus this 0.4554 p-value result of the PDF 1.2. So it shows no significance, so it does not suppress PDF 1.2. So in the future, I will replicate these results to confirm them, and this will allow me to set up an experiment correctly for Arabidopsis thaliana. And I just got to view how plant infection takes place, the symptoms, and the time requiring for it. I would like to thank my research mentor, Dr. Deb, for helping me with this process and just helping me see how amazing plants are. And she taught me so many techniques that we'll use in the future. And overall, I just hope that you guys enjoyed my research, and I can't wait to answer any questions. Thank you.